It's my pleasure to open session 3, in which we talk about nanomedical structures and devices in development today. One of the most important macro structures is our skeleton, and our first spokesperson today is going to be talking about bio-nano scaffolds for personalized post-traumatic osteogeneration. He's an expert to be speaking in this area. His name is Dr. Ennio Tasciotti, I'm sorry.
Nobody gets it right. So.
Dr. Ennio Tasciotti. Perfect. Practice makes perfect, always. Dr. Tasciotti earned a master's in science from molecular biology and a PhD in molecular medicine from Scuola Normale Superiore in Pisa, Italy. His postdoctoral work on gene and stem cell therapy was completed at the International Center for Genetic Engineering and Biotechnology at the Laboratory of Molecular Medicine. He's currently an assistant professor at the University of Texas Health Center in Houston. Dr. Tasciotti will speak today on bio-nano scaffolds for personalized post-traumatic osteoregeneration. It's our great pleasure to welcome Dr. Tasciotti back. He was one of the most appreciated speakers in the reviews from previous conference, so it's a real pleasure to have you back. Yeah.
I always wish my mama could hear these presentations and be proud of me, because you're always too generous. So I'll talk about this fancy name, bio-nano scaffolds for post-traumatic osteoregeneration. As I said, it's a fancy name, first of all, like all the affiliations and the institutions I work for. So it's a very fancy name for a very basic and urgent need, which is try to find a solution for a problem that the Army brought up to us, which is the amputation of the limbs of the soldiers due to shattered bones. So the Army did a great job at saving the lives of the soldiers, didn't do a good job at saving the limbs, and that's what's causing more than 4,000 amputations to these days. And it's incredible, because even if you just have all the tissues around and the nerves and everything, but you don't have the bone in one piece, you still need to amputate a limb, which is kind of a traumatic intervention. Now, we got contacted by this guy in the middle, Mitch Zekin, who is a program manager at DARPA. DARPA is the Pentagon Defense Advanced Research Project Agency, which deals with all these crazy ideas and revolutionary research. This is a picture that we took at the kickoff and review meeting in Key West in January, and there's a reason why I put it there. You'll discover it at the very end. Anyways, when they contacted us, they asked for miracle material, which we call it fracture parity, but this miracle material should do a lot of things. Should provide, it's like the 10 Commandments, provide temporary mechanical support, act as a substrate for osteoid deposition, so to promote bone regeneration. It should somehow mimic the architecture of the real bone, so promote also vascularization and in-bone growth. It should encourage the bone cell migration into the scaffold. It should be osteoinductive, enhance the cellular activity towards osseointegration, degrade in a controlled manner so that the load transfer to developing bone can be taken care of, produce non-toxic degradation products, be non-inflammatory and non-immunogenic, and it can also be sterilized, because it has to be shipped to all corners of the world. So a lot of questions, and of course I can't do not even one of that by myself. So we really ended up putting together a team of stars in the world of science. I just had the pleasure to coordinate the whole team. I wrote the grant and I put together the team and I'm running it, which is crazy because it's like 60 people, 7 different institutions in 5 different cities. But anyways, these are the players. And these are also the components that we're gonna use. We have a polymeric matrix, polymer, polypropylene fumarate, developed by Tony Mikos. This is the guy over here at Rice University, and these are his collaborators. Then the nanoporous silicon enclosure, something that I presented last year, and that's my staff together with Professor Ferrari and a lot of other people like Professor Liu, and that's the rest of my team. Then the peptide amplifier biomimetics, a technology developed by Sam Stoop at Northwestern and by Ramil Capito, who is his assistant professor. Then we have mesenchymal stem cells, because there is no way to promote osteoregeneration and bone growth if you don't put a little bit of stem cells. And we're working with Paul Simmons, who is one of the world experts in mesenchymal stem cells. He's already in clinical trial in Australia for the use of mesenchymal stem cells. And then we had the pleasure also to work with George Whiteside on the development of the bioadhesives and the management of bioinformation, which is very crucial when we talk about bone tissue engineering. And then we have a lot of mathematical modeling, which I will skip. Otherwise, you will all fall asleep. But it's the work done, again, by Professor Ferrari and Professor De Cuzzi mainly. A lot of biomechanics done by the team at the Institute for Orthopedic Research by Phil Noble, Hugh Jones, and the imaging that we developed that's been developed by Raffaella Righetti at Texas A&M. So a very multidisciplinary team brought together with a common objective. First of all, generate a first generation of bio-nanoscaffolds that could do a lot of things. So they could provide for the loading and release of bioactive molecules. They could, with the curable polymers, provide the polymeric matrix that cures in vivo. With the bioactive peptides, something that can generate the right environment for the stem cells to grow, and something that can adhere to the bone end and not to the soft tissue. So this is the matrix that I put together while I was planning and envisioning the grant, and it's in 3 steps. So that's the first in vitro stuff, just the material part. Then all the biology that has to be developed to accommodate the stem cells and make them grow happy. A lot of animal models will be discussed in the presentation. And finally, the third stage in which we have developed everything with the clinical guidance of a team of wonderful orthopedic surgeons. And we have here in the audience one of them, Harvey, raise your hand. Harvey is one of the associates of Brad Weiner, who is our leader in the clinical guidance. So a very complex structure for kind of a simple idea originally, which is develop something that you can inject or mold, something really like a Play-Doh. That was the original idea that I had when I was thinking about this project. Something that you can inject in the part that is missing, so let's say like a gap in a long bone such as the femur or the tibia, that contains different components, and these different components provide all the factors and the environment that is necessary for the bone to heal. So the cells would reinfiltrate the area, will proliferate, minerals will be deposited, and eventually and hopefully the bone will form again. So we have already done like step 1, 2, and 3. We're now waiting for step 4. We still don't know if in the long run all the bone will be made. Despite that the idea was kind of revolutionary, because there is nothing like that, again, Mother Nature developed all of these in a pretty spectacular way. So bones in general can heal, and can heal in a very good way through a series of steps. And so what I did at the time was try to inspire myself from what happens in nature and try to replicate it in the lab. We did a lot of work in terms of biomaterial integration. So unfortunately, I cannot give any of the details because the patent attorneys grilled me for like an hour So a lot of the details, unfortunately, I can't give them. But we did a lot in terms of integration of three materials that never got together before. So amphiphilic peptides, polypropylene fumarate, and nanoporous silicon were never put together. And so we had to develop a lot of chemistry and functionalization in order to make them go together, in order to make them be cell-friendly, because eventually cells have to grow on this stuff. We did a lot of mechanical testing in order to achieve the mechanical properties that are required in order for this material to be mechanically solid. So the idea that Mitch Sakin at DARPA had is that, okay, this is the first thing that he said when he came to talk to us. He said, I want a material that the soldier can inject behind the rock, and after 2 hours he can go back in the battlefield. And he said, yes, to get shot again. That's what I said, because that's crazy. So we managed and we negotiated a little bit back and forth, and eventually agreed, okay, you guys got 2 weeks to have a complete mechanical stability. So you inject this material can stay in the leg of the soldier while the soldier is immobilized for 1-2 weeks max. But after 2 weeks, the soldier has to be able to walk on that thing. And after 6 months, that injectable material that you introduce should be disappeared, and at its place there should be natural bone. So that was that's the goal. That's what I think about day and night. So we developed a lot of mechanical tests, and we started from this was the first one, the first material that we developed. Very poor mechanical properties. We increased it a lot. We're now very happy. We can do different type of cross-linkings, just chemical, temperature mediated, the UV light, so something like the UV light that dentists used. And we're very happy because now this is the final composition. Now, according to the FEA models and according to literature, the maximal stress in a tibia or in a femur is around 20 megapascal, so which is here. And our fracture point is at 70. Which means that we can sustain a 3.5 body weight of the sheep, in this particular case, before this material will crack. So we are kind of confident that the sheep will walk after we take away that 5 centimeters of femur or of the tibia. So that takes account of months of work on the biomaterial development. As I said before, there will never be the good regeneration of the bone if we don't have the right biology on board. And the right biology is driven by the right factors. And so mesenchymal stem cells are the players, but also the mesenchymal stem cells have to be recruited and retained in the scaffold. They have to be forced or stimulated to proliferate and eventually to differentiate in the type of bone cells that you need. So we just developed a lot of different delivery systems, and this is what we're good at UTH. This is the work of my group. So we develop these silicon structures. They're porous. They're very cheap to make. They're very reliable in terms of size and shape, reproducibility. We have different size and shapes. We have different porosities. This is all in the same scale. So the advantage of this is that these guys have pores all the way through the structure, and you can load them and pack them with nanoparticles, growth factors, biomolecules, and once they're inside, I'll show you later, they're protected from degradation. So different sizes of pores according to what you need to load inside, so that they can accommodate from a very tiny molecule, 2 nanometers, to a big liposome, like 150 nanometers. We also develop a lot of different coating strategies, capping strategies to match the requirements of these time-staggered release of the molecules. So now we managed to have releases in terms of hours, in days, or in weeks, according to the different recipes and the different structures. We tested all the different structures, all the different growth factors, in order to identify what was the cocktail, the best cocktail of factors that could promote the cell proliferation. So all this work now pointed out at these final composition, and that's what we're using at the moment. As I said before, the silicon is a great way to deliver biomolecules because you can control the release, but also because once they are loaded inside, they do not degrade. So this is just BSA, it's a model protein, and these are all the degradation byproducts if you leave it as is, without even adding trypsin, just by leaving it as is for 7 and 14 days. Now, if you load it inside the silicon, you don't see those degradation bands anymore, which means that the protein is stabilized. Also, you can control the internalization of target cells so that you can decide if you want or not them to internalize your particles. They completely degrade, they're completely benign. And actually, the good thing is when you combine PLGA and nanoporous silicon enclosures, NSCs, the degradation byproducts respect the neutral pH, which is very important because most of the polymers that are used in the bone tissue engineering, such as PMMA, they say they're biocompatible, but they create a hell of inflammation because they are like the byproducts are very acidic. So that's something that we wanted to be very careful about. So now the amphiphilic peptides, a quick thing. So you need the cells, you need the right materials, and you need the right environment. And these amphiphilic peptides in the right composition, they promote the deposition of minerals. And the good thing is that the deposition of minerals is also very close to the ratios that are typical of natural bone. So natural bone is 1.657, this is 1.65, so very, very close. And you see the way they assemble, they created this dense network of fibers, which resembles very closely the extracellular magic, so cells love this thing. Then let's go to the cells. The cells have to do a bunch of stuff, that they have to stimulate the angiogenesis, they have to recruit other mesenchymal stem cells, they have to proliferate and differentiate, and so on and so forth. We follow two approaches. We use either the cells that can be derived from the bone marrow, so from just a bone marrow aspirate with a few of a little bit of cleaning with fax, or we isolated this new class of cells, the compact bone. So by taking the bone itself, crushing it a little bit, and doing some other magic, which I can't say, but anyways, we isolate these compact bones, and the amazing thing of these compact bone cells is that they grow 40 times faster and better than the normal bone marrow-derived cells. So these cells are, what does it mean?
It means
5 minutes. Oh my God.
But if you want to go longer, there could be less questions.
Okay.
You can talk into your questions.
Okay, I'll go faster. So anyways, they differentiate in all the different lineages, and we also tested the way they assemble on the scaffolds. Now, I'm not sure that you can appreciate that this is like a Z-stack throughout 200 microns of the scaffold. And you see in red the particles, like the delivery system, in green the cells. And you see that they are dispersed throughout. So it's very uniform the way they grow inside the scaffolds, which is again, it was not a piece of cake to get. And you see again in confocal analysis that they never co-localize. Again, you don't want the particles to get internalized by the cells because the particles release growth factors. Growth factors are active outside. If they get internalized, they're useless. And so again, this is another reconstruction, and you see that they're never together, even though they grow throughout the thickness of the scaffold. Of course, another very important thing, the scaffolds need to degrade eventually, as we said, and they have to degrade in the arch of 6 months, because if they degrade too fast, then the bone didn't really develop at the point in which it can sustain all the loads and the forces that act on it. So we had to do a lot of different recipes, and we're now kind of happy because the bone formation starts officially at 1 month. We try now to anticipate it a little bit better, and according to the different formulations, that's when basically the degradation starts. And a long time you see that the scaffolds became more and more porous. You have to imagine now in these pores the bone tissue that has been regenerated that is infiltrating and filling the voids and the gaps. And again, we always keep an eye on what happens in terms of pH, because something as simple as pH can mess up the whole experiment, and we're happy because we keep it around 7.4 no matter what is the composition. We also analyze all the byproducts. The mathematics, I'll skip it super fast. Now, we're doing now a lot of mechanical testing. We work with a lab that has incredible abilities in testing implants and scaffolds. So they developed a lot of tests. Now, this that seems like a big bone, this is the bone of a rat. So it's something like this big. But they managed to develop a test so that we can understand what are the mechanical properties of the materials when we implant it in the bone of a rat. So without touching the sheep, which is of course more expensive and more a little bit trickier to deal with. So they have all these FEA models that they developed. They can merge the micro CT with the mechanical testing so that they see the displacement, the tilting, and this eventually got to the miracle, the dead sheep walking. So basically, even before, that's how we called it. It's a gait simulation. So basically, this is the leg of a sheep, of a real sheep. So we take the whole leg, and they rebuilt it on this machine, studying the different ways the sheep walks. Because we don't want to get to the point of doing surgery on a sheep, and after 1 week, the sheep stands up and breaks the bone. We don't want that. So we recreated this system and we animated the sheep leg. So now, even if it might be kind of gross, but it's very useful for the surgical team because we can also do surgery. These have muscles, tendons, everything in place. It's just explanted. We can put the implants here and check if the mechanical properties will be solid enough to sustain all the loads that can be applied in all the movements that are usually performed by a sheep while it moves, which are not only the bending but it's also like the torsional movements yes, like this is a torsional movement, just in case that the sheep wants to dance a little bit. And this is going to be the final mechanical test. We're now trying to develop all the surgeries on this. Now, the other important thing that you have to consider is that we have so many different components. I always kept an eye on trying to develop something that could be put in a kit so that the surgeon in Iraq or in Afghanistan opens it and he finds all the different ingredients. The aliquoted MSCs ready to thaw, the sterilized, sealed, pre-treated scaffolds, the lyophilized NSCs, the PA mixes. You just have to put everything together. We develop a proprietary protocol to gel everything on the scaffold. And this is the first experiments on the bone of the rat. So this is a very tiny bone. We applied this screw because the animal committee didn't trust that we could recreate the bone, and so we have to do that. But then in this void space, we put the injectable putty and we test that everything works fine. Now, I said it's injectable, and I wanted to actually show it to you. So this is one of my students putting together the injectable putty. He's putting it in a syringe. The syringe is now loaded. You can, in this case, we're injecting just in a mold, in the mold that we use for the mechanical testing, but you can imagine to have a needle and to inject throughout the skin. We're now doing these experiments. So you put it here, and then you put this whole thing in the cell incubator for 15 minutes, so at 37 degrees, humid condition, doesn't matter. And after 15 minutes, you go back, you take it, you push it out, and this thing is now a solid cylinder with very strong mechanical properties. So it cross-links in vivo at 37 degrees in 15 minutes, and it's very solid. And also, of course, it can be shaped in any size you want according to the bone that you're working on. So that's why I was talking about a Play-Doh. So if you have a long bone, you have a long bone. If you have something like this, should be a vertebra, according to my student. Of course, it's not a good sculpture. And this is like the results. So I want to show you the different implants. I want to show you that after 4 weeks, you have a complete integration of the scaffolds with the surrounding tissue. You see vessels growing in, vessels going out. And if you start doing the histology, you see a lot of the classical features of a natural bone. You see osteoid, you see osteoclast, and interestingly, you also see osteoclast. Osteoclasts eat bone, so they have no other reason to be there but to eat something. So it means that they actually found bone to eat. And also you can see like infiltrating blood vessels, which are necessary. Now, I don't have time to go through all the different groups that we tested, the different combinations. This is like a lot of work. I just want to leave you with this final picture, which is a color coding of the bone that has been formed ex novo on the scaffold. So where you see green, this is the new bone that after 4 weeks only has been formed in the scaffold. And again, we're trying to do even better. Conclusion, identify the growth factors, the molecules that are needed. We got to the right mechanical properties. We assessed the biodegradation, evaluated all the activities of the stem cells, incorporated all the materials together so that they go together very well, and develop all the in vivo stuff. So again, I really want to acknowledge all the work of this incredible team that I have the pleasure and privilege to coordinate. And the reason why I put that picture is that we did this January the 5th in Key West, and this was the weather. Maybe next year, you might want to consider Florida.
Okay.
Just an idea, just an idea. Thank you.
We have time for 1 or 2 questions. I think you knocked us all dead. Okay. Oh, one question.
So, Chris Schaffmeister from Temple University. So you showed degraded BSA without proteases or anything?
Without, that's just temperature. It stayed there in 2 weeks. So
and there's nothing growing on it or anything?
No.
So is it getting chopped up or aggregated?
It's getting chopped off. So
Okay.
we're now doing all the HPLC and MALDI characterization to understand exactly what are the molecular species. I just talked about one of the the graphs, the silver stains. The other silver stain had the trypsin added. So we're doing both. So now if you look at this one has a oops. Okay, can you put back the, yes. You see this as with trypsin in 10 minutes, and you see that the degradation bands decrease, so you can still prevent. And the reason is that the pores are small enough so that trypsin cannot get in.
So this is when it's in the matrix, and then
Yes.
on the right is when
Yes,
it's not in the matrix.
just in solution.
Okay, thanks.
Thank you. Okay, I think all of you will join me in agreeing that Dr. Tasciotti gave us a fantastic presentation, and we owe him a trip to Key West for next year.